Hello, everybody, and welcome to our research paper review series. It's our second episode in the series, and today we're going to review one of the papers that we just got online, High Confidence Cancer Patient Stratification through Multi-Omics Investigation of DNA Repair Disorders by Garrick Mercadotechan. And of course, here we want to highlight that it's not the Insilico paper. It is the paper which originated at the University of Copenhagen at the wonderful lab of Professor Morten Scheibe-Knudsen, MD, PhD, who also is the lead chair of the wonderful 10th annual research drug discovery, aging research and drug discovery meeting. It's the anniversary, so please do come to the conference. And if you haven't heard about it, please go to agingpharma.org and register. The event is epic. All the big pharma is coming there, academia, really top experts. 41 weeks to go, and we already have 35 speakers confirmed. Several sponsors. By the way, don't forget to sponsor. But back to the paper. Again, want to highlight that we are co-authors, but not the main drivers. And in this paper, we've utilized the power of artificial intelligence for patient stratification and also target discovery. And I would like to pass the word to our head of Insilico Hong Kong and head of the applications team, who is leading a team of wonderful target discovery sciences utilizing PandaOmics platform. Dr. Frank Pan. Frank, please lead the discussion.
Okay, thank you very much, Alex. I think it's really nice to have the main contributors of the paper joining our today's discussions. Well, maybe before we go to the detailed discussions, how about Morten, Evgeny, and Alexandra briefly introduce yourself and your backgrounds? Maybe we can start with Morten. Thank you.
Thank you so much, Frank. Yeah, so my name is Morten Scheibe-Knudsen. I'm an associate professor at the University of Copenhagen, and I run a research group where we try to understand aging and diseases associated with aging, such as cancer, and what we can do to best treat these disorders.
Thank you very much, Morten. Evgeny, please.
Thank you, Frank. Great to see you here, guys. My name is Evgeny Izumchenko. I'm assistant professor at the University of Chicago. My group is working on head and neck cancer for many years, and for almost a decade, we're using tools developed by Insilico to analyze the very complex driver that drive head and neck cancers. We published our first paper in 2012, and this collaborative study is our paper 25th. And I'm looking for much more to come, especially since we have established this multi-institutional group, which obviously led by our colleague from the Copenhagen.
Thank you very much, Evgeny. And Alexandra, please.
Thank you, Frank. So my name is Alex Deviorsky. I am a bioinformatician at Insilico Medicine, and my main role is to coordinate and provide bioinformatics support for both our external and internal programs. And actually, the first project that I was assigned for at the very beginning of my career in Insilico Medicine in early 2018 was a collaborative project with the University of Copenhagen, so secretly called the DNA Damage Response Project. So a lot of experiments were done since that time, and I hope that world will hear from us several times again very soon.
I see. Thank you very much, Alex. Well, I know you put great efforts into this paper. Could you please briefly highlight the key findings of the paper for us? Thank you.
Yes, Frank, of course, but before a brief introduction to paper, I just wanted to highlight one of the problems in target discovery that usually occurs. So there are plenty of targets for certain diseases with strong biological evidence behind them, but for some reasons, these targets don't reach clinical efficacy. So One reason might be due to poor recruitment strategy, and it highlights the significance of the usage of molecular biomarkers to improve patients' selection criteria. And it's also commonly known that one of the important trigger of cancer predisposition is mutations in DNA damage response genes. So we assume that analysis of the DNA repair disorders with high cancer predisposition and identification of cancer predisposition signature might be a good starting point for the investigation of molecular biomarkers for cancer. And as I already mentioned during my introduction, so we have a collaboration with Morten's lab, super experts in DNA repair disorders and DNA damage response in general for more than 5 years. And we also work a lot with Evgeny, an amazing expert in the cancer field. So since the time we at Insilico Medicine developed PandaOmics, a powerful tool for multi-omics data analysis and target discovery. So just putting all these factors together actually resulted in this article. So in general, this article provides an example of how the integration of multi-omics into target discovery pipeline might be beneficial for the development of tailored therapies, and underlines the importance of the identification of molecular biomarkers for cancer patient stratification. And just a few words regarding this paper. So what was done? So for the analysis of DNA repair disorders with cancer predisposition, we used PandaOmics platform for the identification of cancer predisposition signature, and found a link between the expression and of those genes and survival outcome of cancer patients that allows us to hypothesize that some of them might be used as predictive biomarkers for patients with poor survival, bad prognosis. So next we focused on a group of sarcoma patients with bad prognosis. And to do so, we applied PandaOmics based target ID algorithm and identified several target strategies for this group of sarcoma patients with bad prognosis. And among them, we identified PLK1 as a potential target and tested it in vitro. I think that it is important to mention that PLK1 was recently proposed as a target for treatment of sarcoma, but potent PLK1 inhibitor showed limited anti-tumor activity in recruited patients. So presumably pure recruitment strategy was the reason that the trial did not reach its efficiency endpoints. And as I already mentioned, it highlights the importance of the identification and usage of molecular biomarkers for stratification of cancer patients.
I see. Thank you very much, Alex, for the great highlights of the paper. I can see that it is a really nice paper. Well, early this year, we used the PandaOmics to identify the dual purpose targets for aging and age-associated diseases. And this summer, we used the platforms to identify 8 unreported targets in ARS and further validated them in the FRI model. I'm really proud to see another demonstration of the platform with different practical settings that really allow us to analyze massive data sets and then to find out the novel markers capable to stratify the cancer patients with different survival outcomes. And then finally, we can also find the therapeutic targets. So when we are talking about the patient's stratifications, may I know how important it is, or what are the major problems with the existing therapeutic strategies for treating the major cancer types for all patients? Well, I guess, Evgeny, I think you are the best persons to answer these questions and comment on that. Thank you.
Yeah, Frank, thank you. Great question. This is the question number one in the cancer field, right? How do you recruit the patients to the trial that you assume are most likely to benefit from the trial itself, right? This is the question number one in cancer, because most of the trials fail just because of that, right? So patients with exactly the same cancer, histologically and clinically the same cancer, they have different biology that drives the cancer. That means that the single drug will not work for all. So the field goes towards recruiting the right patients with the right molecular profile to the trial, which we hope and we know that it will increase the chances that a larger number of the patients in the trial will respond to the treatment. And this is exactly what this paper shows. This paper shows that you have a very creative, a very, I would say, secure way to stratify the patients with novel biomarkers. The field of the cancer biomarkers, it's pretty old, right? So some of the cancers, we know the biomarkers for quite a long time. Now, the thing is that, first of all, most of the biomarkers, or definitely not all of them, are functional and targetable. That's one thing. You know that you can use a molecule to stratify the patients by the survival or an outcome, but not necessarily you can use this molecule to drug, right? And second, there are a lot of new biomarkers that we absolutely know that they're out there, but we cannot detect them because, you know, what field is doing over and over again, you just compare the patients to healthy individuals and you try to find what separates them. What was done here is very different. Instead of going directly to the cancer patients, right? What was done here is pretty smart. This group first went to the patients with non-cancer disorders, but with conditions that are driven by defect in DNA repair. We all know that dysregulation of DNA repair machinery is the signature of all cancers, right? But by going into cancer patients, we will find the same molecules again and again. What was done here different. We went to the patients which don't have cancer, but they do have dysregulated DNA repair machinery. Identified what makes those patients different, because those patients have higher predisposition to develop cancers. And then we use this knowledge to apply to a large data set of cancer patients and to find how these changes stratify the patients. This approach was not done before. Now, friend, just 1 minute back to your questions. This is exactly what the field potentially needs, right? We need to find a way to stratify the patients, not with biomarkers or the set of markers that we know. We need to find a way to stratify the patients based on new biomarkers that not yet know and not yet have been developed. And that's why this paper, it's a strong leap, not just for the patients with a sarcoma. This strategy can easily be applied to any cancer and non-cancer pathologists, right? Because most of the tools that Insilico working on, they are pathology independent. They can be easily adjusted and adapted to study aging and ALS, cancer, even COVID. This was shown by many, many papers that the group published over the last 2 years.
Thank you very much, Eugene. I totally agree with you. I truly think that is the beauty of the paper. This is a very great strategy. So my next question comes to Mortens. So, may I know, how do you come up with this brilliant idea to choose this approach? For example, to use the DNA repair-deficient disorder to find novel biomarkers for those cancer patients?
Yeah. So, I mean, this is something that we worked on for quite some time. The idea is to stratify diseases based on the phenotypical traits. And so we know the molecular basis of many of these diseases, but we don't necessarily know the reason why phenotypes develops. And so there was a slight correction to Evgeny's point here that diseases that we clustered, some of them do have cancer and some of them don't. And so the 3 diseases that we analyze share cancer phenotypes, but they also have all kinds of other phenotypes. So there's a large phenotypical variety, and the overlapping here was that they show cancer phenotypes. And so that means that whatever is overlapping between these 3 disorders in terms of dysregulated gene expression or other pathologies are probably somehow related to cancer. So this is how we started.
I see. So a follow-up questions, do you think we can apply the same approach to other cancer types or even different types of diseases? If yes, do you have any advice for that?
Yeah, so this is something that we can definitely do. It's something we've actually done before, also looking at how certain diseases show overlap with diseases where you have defects in mitochondria, for example. They show strong neurodegenerative phenotypes. So we can also use it to stratify syndromes where there are other defined phenotypes, and then find associations between these genetic diseases. I think the importance here is to use monogenic diseases, so diseases where you have single genes that are driving the phenotype. That means that you're relatively sure that that gene is important, and then you can find overlap between these very complex syndromes. So this is definitely an approach that we can do for actually any type of phenotype that we can think about.
I see. Thank you. So, Eugenie, do you have anything to add? Because I know that there are different types of hallmarks of cancer as well. So, well, other than DNA repair deficiency, do you think any other hallmarks can perform much better?
Of course, one of the hot topics right now is the background of the cancer, right? And that's what we're doing together with Insilico for many years. We published a couple of papers on the topic, and I do believe that applying the same strategy will definitely increase an insight and knowledge of how the immune system is involved in escape of the tumor cells from the effect of the immune system. Especially what we're doing right now, especially it may find the very specific set of biomarkers that allow the precancerous cells to develop to an invasive cancer cells, right? And by simply comparing the premalignant cell to the tumor cells, it will definitely identify the set of biomarkers, but it's not enough. You will not see everything. So applying a different strategy, exactly like was highlighted right now by our colleague from the University of Copenhagen, may identify the fine-tuned biomarkers that will be masked or missed when you simply compare the normal patients to cancer patients, or when you compare precancerous cells to cancerous cells. So exactly the strategies may show you what are beyond the simple comparison between the major phenotypes.
I see. That's very inspiring. Okay, so what are the next steps? So do you have any plan for those identified targets and the biomarkers in these studies?
Obviously, this is just the beginning, and we have a large collaboration going with Insilico Medicine that have been ongoing for several years, and we have great plans in the works, and I'm sure that we will we'll hear about them very soon.
Oh, yes, of course. And as we all know that even the PandaOmics is only the first part of our fully integrated pharma AI platforms. Okay, so once we identify the targets, we can further use the chemistry 42 to design the novel molecules to tackle that targets, and also use in clinical to predict the clinical trial outcomes. So yes, we can definitely follow up with those great targets and biomarkers. Yeah. Well, as you mentioned, we have a serious collaborations. Well, I'm curious, I guess many of us would like to know, how was this amazing collaboration was formed at the very beginning? Yes. Could you tell us more about that?
Shall I, or will you, Alex?
Sure. So it's a fun story. It started, it's a very small world in AI-powered drug discovery, but even a smaller world in aging research and drug discovery. So one of the interns that joined Insilico in 2014 was Michael Petter. And he was also at that time a visiting post-bachelor's scientist at the National Institute of Aging. So he had kind of two hats. In the daytime, he was working in Morton's lab, and in the nighttime, and sometimes also daytime, he was working in at Insilico, and he wore again two hats. Mostly, he was in charge of, believe it or not, PR and communications. And at that time, we didn't really have anybody dedicated to that. And one of those days, he's like, Alex, you know what? You should meet my boss, Morten Schreiber-Knudsen. And I'm like, who? And it was a very long name, so it took me a while to remember it. But in 2016, that was the first time when we actually physically met. So I still remember that our first beer at the marina in Baltimore, and that became, the start of a wonderful friendship. Morten at that time was moving to Copenhagen, and surprise, surprise, he was also taking Michael with him. So they moved to Copenhagen. It took them maybe a year to set up. They also started doing quite a bit of work in machine learning and AI, and hired one of the, actually the first author of this study, Garik Mukherjee, who had at that time already pretty sophisticated bioinformatics skills, and had a PhD in bioinformatics, to my memory. And he also was pretty familiar with our algorithm. So when he started, he, of course, took maybe a year to set up the experiments. And of course, it's, nothing in academia is as fast as in a startup, but they actually do a lot of work on the planning and grants, etc. So it's very grant dependent. And Garik started working on target discovery, target validation. And I hope that this is the first paper in series. And of course, I don't want to steal the thunder, but there are, of course, many targets that we've identified through PandaOmics that were validated by Morten's group. And, in some companies, like, for example, in AstraZeneca Benevolent, they partnered on target discovery. It took them 3 years to get to early stage drug discovery pipeline R&D. For us, we took, I think, about maybe 1.5 years to validate the targets that were pretty crazy at that time. So completely novel and unexpected. So I hope that this will continue as a collaboration. And I'm extremely excited also by the fact that Morten originally became a co-chair, and then he took over the conference, ARDD. It's now the world's largest conference, and we often call ourselves the Copenhagen Mafia because we are Copenhagen Longevity Mafia. Because it's such an extended group. Everybody goes to this conference, collaborates with Morten, very often with us as well. And it's a truly amazing event. Even high school students now go there, and they use PandaOmics and they validate the targets. That's pretty crazy, right? That's what I'm extremely happy about. And we are now very closely collaborating to expand this field. And I am deeply, deeply honored to be collaborating with Morten's group, which is currently on the meteoric rise. So by the way, if any grant makers are listening, that's where you need to put the grant money, because just put a little bit of money in there, and those seeds grow. They cannot just stay in the ground. So this first paper is a very good example. Also, if you like to learn a little bit more about the paper, about the collaboration, there is a wonderful resource called Nature Bioengineering Community, bioengineering.nature.com. You will be able to find the behind the paper article about this collaboration, about this paper. Just to conclude, would like to pass the word to Morten, who is again the superstar industry we deeply aspire to, and deep bow for making this paper happen, and hopefully we'll be able to publish the rest of the papers on target validation. Thank you.
Thank you so much, Alex. And it's been a long journey, and I'm really excited about this first paper, and also really excited about our continued work together, in particular with you know, finding novel molecules that we can use for targeting some of these diseases that we are fighting against. So really appreciate all the years that we've worked together, Alex, and also Alex V. It's been a pleasure to working with both of you. We have
and Evgeny.
And Eugenia, of course. But Eugenia has joined a little bit more recently, but it's been extremely and a great pleasure to work with you, Eugenia, also, of course. So much energy and a great insight, and I really appreciate all of this. This has been fantastic. So thank you so much for all your support and help with this.
Thank you so much.
Yeah, Evgeny, please.
What I wanted to add is that it's amazing to see how experts from different fields get together around Insilico. It's like a magnet that brings experts, and experts stay there. And they stay there because they see that what Insilico is doing and the tool that they are working on, they're multifunctional. And that these experts can use them again and again as these tools grow, evolve, and get better. I started to work with Insilico Medicine in 2012, and as I mentioned in the beginning of the session, we published 25 papers together. So I was using the tools that Insilico was working on cancer. But of course, agent, it's a major field that the company is focusing on, and Morten, of course, became quickly the essential part of this branch, right? And I think it's just natural for us, right, over time to get together and start working on this line that connects cancer and aging, because we all know that cancer, it's aging condition, is a pathology that associated and driven by many pathways and molecular signaling that is involved in both, right? The same targets may potentially be targeted in aging and in cancer. So this is amazing to see how different people with different focus, my lab on cancer, Morten on aging, you know, how by working independently with Insilico, right, we started to publish, and then we eventually come together and we're starting to use an expertise that we have accumulated over years to solve new problems, right? New old, right? And I don't hope, I know that the work that we just started with Morten will continue. We have many more data that we're not showing in this paper, but definitely it's soon to be published. So thank you, Alex, for being the magnet for multiple experts for many years. And I think that good people stay.
Thank you, Evgeny, for those kind words. And for all of you who are still listening, in the case that there is a chance that somebody is here, ARDD, join the University of Copenhagen conference. It's a 5-day event, and join the Copenhagen Longevity Mafia. We work in the day and at night to make people live longer. That's unhealthier. So thank you very much, and deep bow to everybody. Yeah, and cancer-free. Yeah.